We do currently pursue two portfolio programs targeting, in fact, microglia in the context of neuroinflammation. One of them is TREM2 agonism, the other one is Kv1.3 blockade, and both of them are maturing very fast through the two platforms that I've just described. We are testing both tool compounds as well as proprietary compounds in vitro and in vivo. From the all-in-human discovery platform, we are, of course, particularly interested also in identifying biomarkers, which are essential in the field not only of neurodegeneration and the identification of patients at the right disease stage, but also to identify biomarkers that are tracing neuroinflammatory processes as well as that monitor target engagement. We are at the same time building the structural information around the targets themselves that allow us to fine-tune the structure-based drug design to modify the compounds further. And at the same time, we are utilizing the spatial transcriptomics platform in order to identify novel target candidates. And here the exciting news is really that we could show by comparing resilience towards pathology with Alzheimer's disease that indeed spatial transcriptomics suffices to identify not only differentially expressed genes, but also clusters of genes that are mapping onto biological pathways that are showing a clear differentiation, for example, in context of proximity to pathology, comparing AD with resilience, and that then brings us further to the identification of particular members of the biological pathways that could function as drug discovery targets. And here we have now identified a handful. Some of them actually map onto biological processes that have already been brought into the context of neurodegeneration, like, for example, mitochondrial health, but also microtubule function. So highly relevant biological processes with regard to neurodegeneration, where we now have novel entry points through differential expression of gene factors with that point us towards potential new drug targets.